Hi, everyone. So I'm here with Jae Young Lee, who is a lead scientist at ToolGen. ToolGen is a company in South Korea who's working on a gene editing program for CMT1A. I'm really excited to have you here, Jay, to tell us a little bit about your program. So talk to me about the CMT program that you guys are working on.
Thanks Susan for introduction. Our approach for the CMT is to edit the root cause of the disease, because CMT is driven by the genetic mutations, and as some of you may know, there are several or 60 or 70 mutations out there that can cause CMT. And we first and foremost, we're trying to target the type 1A of CMT, which is driven by the mutation in a gene called PMP22. And we are trying to target the root cause, which is PMP22, by gene editing tool.
And tell me, how did you all select CMT1A versus all the other mutations that are out there?
There are many mutations that can cause CMT, but in terms of the prevalence and incidence wise, CMT type 1A it covers about 50%, almost half of the whole CMT patients. We're trying to target the CMT1A first and foremost to get the as many patients that we can target, that we can deliver some treatments for the patient community.
That's really interesting that the prevalence is driving the technology. Obviously, your technology is well suited for CMT1A. Will you talk about that a little bit?
Yeah, sure. We are dealing with the gene editing tool. It's called CRISPR-Cas9, which is developed in 2012. There have been many gene editing tools out there, but unlike other gene editing tools, CRISPR-Cas9 is relatively easy to handle. And believe me, if you are not a molecular biologist, but even if you're not molecular biologist, if you're with me about a month, you'll be able to handle the gene editing very easily. So that's how easy it is. And we're trying to translate this CRISPR tool to clinical novel therapy. And for CMT1A specifically, as some of you may know, PMP22 duplication, so there's an extra copy of the PMP22 gene drives the CMT1A disease. And because there is a PMP22 extra copy, there is the expression of this PMP22 gene is highly enhanced or upregulated. So what we're trying to do is to target the molecular switch of the PMP22 gene in an attempt to lower down the PMP22 to normalize its expression to get a therapeutic benefit for the patients.
And tell me about your project right now. Where does it stand, and what do you expect in the near future?
Yeah. Well, our CMT program is still at its preclinical stage, where we achieve therapeutic efficacy in at least in some of the relevant animal models. Right now, we are trying to develop better delivery tool to deliver our CRISPR components to the peripheral nervous system, where CMT, where there's a pathology of the CMT comes from. So right now we are trying to develop, like I said, the delivery tool for the CRISPR components, and we're trying to apply that into the animal model.
And how do you envision this CRISPR technology getting delivered? Do you imagine it being a shot? Do you imagine it being IV? Do you really not know yet?
Well, it's difficult to speculate at this moment because we are trying to investigate a lot of options for now. So it could well be a intravenous,
Mhm.
or it could well be intrathecal, which is directly injecting into the cerebral spinal fluid.
Mhm.
Um, but one thing for sure is that our approach will be a single treatment regime, so that patient won't need to visit clinic frequently.
Yeah, that's great. So you only have to go in one time for the treatment, and theoretically, it'll be done.
Yes, yes,
Yeah.
that's true.
So, what do you expect, or what are you seeing in the animal models now as being the result of this CRISPR technology? Are you seeing an improvement in their symptoms? Are you seeing just that their progression stops or it's slowed down? What are you seeing?
So at this moment, we administered CRISPR components to the one of the animal models of CMT1A, which is a rather very severe model. So and we administered CRISPR components to the animal models at before the onset of the disease, and also at the right at the onset of the disease. And both of the time points resulted in amelioration of the neuropathy, and also it means that it improved the myelination, because demyelination, which is damage to myelin, drives the CMT1A, and also it resulted in a improvement in conduction speed of the peripheral neuron.
So that sounds pretty exciting if you're talking about amelioration of the neuropathy. We understand that this is animal models, and we understand that this is an early point of injection, and so or administration. So certainly these are things to take into consideration. So talk to me about where you are right now in the preclinical stage and what breakthroughs or milestones you're going to look for in order to get into the clinical stage.
Yeah. So as I mentioned, although CRISPR component has a great outlook for the future, the major hurdle that we feel is the delivery of the component to the specific body that we need. So right now we are investigating a lot of options that can deliver CRISPR components to the body, and specifically for CMT, delivery into the peripheral nervous system specifically is almost important for us for now. So the major milestone for us would be finding a right and efficient delivery vehicle that can deliver CRISPR components to the peripheral nervous system.
So, all right. So that sounds like a challenge that you guys need to work through, but certainly not a unique challenge to CRISPR. We know with gene therapies, getting into the peripheral nervous system can be a challenge for everybody. And I know it's across the board. That's a big challenge that many different efforts are trying to resolve. Um, when you are thinking about some of your next steps and you're thinking about the clinical application of your technology, have you already been in touch with some of the regulatory agencies in Korea or in Europe or in the United States to understand what kind of an appetite they might have for CRISPR technology for CMT?
Okay, so in Korea, first and foremost, because CRISPR-2 is relatively new technology, so we feel that the regulatory bodies in Korea, at the very least, are not so ready uh, to regulate these novel technologies. But we are trying to first and foremost educate the regulatory bodies to for their for their better understanding of the tool and some of the potential side effects that can be harbored with the new technology, so
Mhm.
that the regulatory bodies will have the right essays and will have the right recommendation for the developers like us. But this is the case in the Korea and in the United States. I know that at least some of the biotech companies are trying to develop the CRISPR components, and I'm sure they had better communications with the US FDA. And for example, Editas Medicine already administered gene editing components, which is CRISPR, to the patients with genetic eye disease, which is the first clinical trial for CRISPR, and we're really eager to find out the results as well.
Excellent. That's certainly something that we'll be watching. And you know, CRISPR is a well-used term. I think a lot of people in the scientific world certainly know what it is, but I think a lot of patients have heard of CRISPR. There's, you know, been a big report on CRISPR being used in mosquitoes to help with the spread of Zika virus. And we're talking a little bit about obviously CRISPR to make animal models like the mice and the rats that have the diseases. That's been used for years. And now being able to translate that into humans is an exciting opportunity. So Jay, what can you tell me about how this kind of technology might translate to people who don't have CMT1A and why maybe they should be excited about the work that you're doing?
Okay, so at least a lot of CMT harvest similar pathological hallmark, which could be demyelination, myelin damage,
Mm-hmm.
or peripheral neuropathy, which is driven by the peripheral neuron or the peripheral Schwann cell.
Mm-hmm.
So Schwann cell is the cell that generates myelin. So as say if we success in the CMT1A with the gene editing tool, we can theoretically target the other gene of interest. Well, it could well be CMT1X, or it could well be CMT2A. I mean, the mutation and the disease-causing genes are known for these sort of types of CMT. So theoretically, we can develop a gene editing strategy for these other CMTs. And given that if we were success in delivering CRISPR-2 to the peripheral nervous system, then we can simply apply that to the other types of the CMT. So we'll have to see how it goes, but we're trying our best at the moment with the CMT1A first. CRISPR components can be divided into two components. So one is being that the eraser,
Uh-huh.
so which is the Cas9 components, which can actually cut the DNA of interest.
Okay.
But this can be guided by the guide RNA, which
Okay.
is basically a GPS for the Cas9 components. So the guide RNA finds the DNA of interest, and the Cas9 can cut the gene of interest. So theoretically, we don't have to change the Cas9 components. We
Yeah.
just have to change the RNA components, which is relatively easy. And this could be a significant benefit for the CRISPR, where if we know the gene of interest that we want to target, from the discovery to the lead compound identification would be very, very short when
Yeah, so
compared
it
to the
just is
other
going to take
modalities.
it's going to take one in order to be effective to then start this snowball effect for all the other types.
Yes, that's true.
Yeah, that's really exciting. And you know what I noticed in that you said, you know, targeting the gene is actually relatively easy. And
It is relatively easy. I mean, believe me, Susan, if you're with me about a month, you'll be a CRISPR expert.
And isn't that exciting? I mean, 20 years ago, we had no idea how to do this. And now we're saying it's relatively easy and we can get into the peripheral nervous system. You guys are working on this really wonderful proof of concept with CMT1A, and it could have wide application for the entire CMT community. I think that really provides a lot of hope to the CMT community, just even seeing the development of the technology over the last 20 years and seeing how what a great opportunity this is and what an exciting time this is for CMT. And what can patients do to help you get there faster?
That's really a good point. And this kind of patient's view, it's really a patient view and going into the clinic, how often and so on. So I believe that we really need the patient's view on the ideal therapy and especially patients' communities input right at the beginning of the therapeutic development would be very highly beneficial for both industry side and also for the patient side for better outcome of the therapeutic development.
Sure, to make sure that you guys are getting the results from the treatment that the patients want.
Yes.
Sure. Okay. Well, count on the CMT Research Foundation to be your partner. And if you need our voice as patients or scientists, we're here to help you. We're really excited about the work that you're doing, and we look forward to working with you and watching the results that you all have and being your partner through the development of it. So thank you.
Thank you very much. Thank you.